Structure of central nervous system. Central nervous system is composed of gray and white matters. Gray matter contains neurons, neuroglia, capillaries, and unmyelinated nerve fibers. Gray matter is present in the central H-shaped area of the spinal cord, in the cerebral and cerebellar cortex. The white matter, on the other hand, consists of bundles of myelinated and few unmyelinated nerve fibers arranged in tracts, neuroglial cell bodies as fibrous astrocytes, but mostly oligodendroglia that produce myelin sheaths in the central nervous system, together with a few blood capillaries. The white matter lacks nerve cell bodies of neurons, synapses, and dendrites. Definitions. Certain definitions are present in the central nervous system. Tract is a group of nerve fibers in the central nervous system which have the same origin, the same termination, and the same function. Tracts are short or long, ascending or descending. A bundle or fasciculus is a group of long ascending and descending nerve fibers which have different origins, terminations, and functions. A funiculus is a very large mass of nerve fibers. A nucleus in the central nervous system is a group of nerve cells in the central nervous system lying very close to each other and having the same function. A ganglia is an aggregation of nerve cells and fibers outside the central nervous system, as the spinal and autonomic ganglia or sympathetic ganglia. A relay station is the synapse, which is an area of contact between neurons. A pathway is a chain of neurons transmitting excitation from one part of the body to another, or from one part of the nervous system to another. A decussation is the intersection of the fibers which cross the midline from one side with those crossing from the other side. Commissure is a band of white or gray matter connecting a part of the central nervous system on one side with the same part on the other side, as the gray commissure of the spinal cord, connecting the two halves of the gray matter. Levels of the spinal cord. The shape of the gray matter depends on the level. Ventral horns are widest in cervical and lumbar segments, in which multipolar anterior horn cells innervating the upper and lower limbs are located. Ventral horns are thin, narrow, and parallel in thoracic segments. Posterior horns are thin and diverging in cervical segments, thin and parallel in thoracic segments, thick in the lumbar segments. Lateral horns are present in thoracic and upper lumbar segments. The amount of gray and white matter in the different segments varies. Large amount of white matter and little amount of gray matter in cervical segments, less amount of both white and gray matter in thoracic segments, little amount of white matter and large amount of gray matter in lumbar segments. Nerve cells in the posterior horn are generally smaller and are of two types. Golgi type 1 are cells with long axons. They belong to the sensory system and are grouped in little aggregations called nuclei. Golgi type 2 are cells with short axons. These cells are association neurons whose axons end either in the same segment or in adjacent segments. Each nerve is attached to the spinal cord by two roots, an anterior or ventral root, a motor, and a posterior or a dorsal root, a sensory root containing ganglia. When the dorsal root fibers enter the cord and the ventral root fibers leave the cord, they lie in the subarachnoid space between the arachnoid and the pia mater. When the root fibers exit through the intervertebral foramina, the arachnoid matter disappears quickly, and the dura mater continues over the root fibers as epinurium.